A pleasant afternoon to everyone. I, Bindu Norona, Assistant Professor, Department of Chemistry, will be the MC for today's program. I would like to extend a very warm welcome to all of you to our 3-day international webinar on pharmaceutical chemistry and intellectual property rights organized by Department of Chemistry, St. Philomena's College in collaboration with Visvesvaraya Trade Promotion Center, VTPC, under the aegis of Department of Industries and Commerce, Government of Karnataka, Bengaluru, India. The webinar on pharmaceutical chemistry and intellectual property right emphasizes on broader goal of drug discovery and optimization. This event aims to expand and integrate knowledge by providing insights into the latest developments and innovations in the field of medicinal and pharmaceutical chemistry. It also covers a critically important session on intellectual property rights. IPR is a prerequisite for pharmacy companies for identification, planning, commercialization, and protection of invention. Hence, the webinar is aimed to create first-level IPR awareness among the teachers, students, research scholars, and young intellectuals about the beneficial aspects of patenting their innovative, creative ideas. The internationally acclaimed scientists from USA and London are invited to deliver lecture on various fields of medicinal chemistry and pharmaceutical chemistry, on the aspects of role of fluorine compounds in the medicinal and pharmaceutical chemistry, drug metabolism, challenges faced by COVID-19 frontline warriors. One song can change a moment. One idea can change the world. One step can start a journey, but a prayer can change the impossible. Let's commence today's program by invocation to seek the blessings of Almighty.
Dear God, thank you for the opportunity to meet together. Please help us to come together to make this institution reflect your kingdom. Breathe life into our ideas and decisions. Help us build a team that has love and respect at its heart. Give us the strength to continue working for your kingdom in this time of pandemic. Lord, come give us the inspiration to be the best we can be. May we be a shining example of your goodness and truth within, wherever we are. Inspire our thoughts, discussions, and ideas, and continue to remind us that all that we do here today, all that we accomplish, is for the pursuit of truth for the greater glory of you. We ask these things in your name. Amen.
With the blessings of Almighty God, I would like to welcome our guests. It is a privilege for me to welcome all the dignitaries present here. I now call upon our beloved principal, Dr. Ruth Shantakumari, to formally welcome all our guests.
A very pleasant afternoon to all people who are associated with this 3-day international webinar on pharmaceutical chemistry and intellectual property rights. This whole body of any organism is a bunch of molecules or chemicals. Being in health or not is dependent on the equilibrium of these molecules, and hence the best method to solve our health problems is to manage the equilibrium of these molecules, and that's what pharmaceutical industry does. So pharmaceutical chemistry is an ever-evolving field, answering newly erupted challenges just like the COVID-19 situation, the pandemic situation, which is bothering mankind now. The other aspect of this webinar is intellectual property rights. Any industry interested in research and development will have to take care of intellectual property rights, protect their inventions, research and development activities if it has to sustain. Unfortunately, most of us, including academic institutions, don't have much idea about what is intellectual property rights, how to protect them, what is the need or importance of patent taking, all these things. And it's apt that the Department of Chemistry and Philomena's College has taken this opportunity. They have identified the gap and arranged this 3-day webinar, and I congratulate them for that. It's my pleasant duty to welcome the dignitaries and all the people who are associated with this. At the outset, we have invoked God's presence for this webinar and blessings. Let me take this opportunity to welcome Monsignor Reverend Leslie Morris, who has been with us spending his valuable time, and here he is going to give the inaugural address. I welcome you, Father, on behalf of the organizing. Professor K. Mahadevan, former Registrar Evaluation, University of Mysore, is going to give the keynote address. And in spite of his busy schedule, he is here to share his knowledge and expertise in the field of chemistry with us. I welcome Professor K. Mahadevan on behalf of St. Philomena's College and specifically the Department of Chemistry, who are the organizers of this program. Welcome you, sir. And then Srimati Prabhavati Rao from Visvesvaraya Trade Promotion Center is here with us, amidst us, and she is going to give the introductory remarks, and I welcome Madam on behalf of St. Philomena's College. Welcome you, Madam.
Thank you so much. Can you hear me, ma'am? Am I audible?
Yeah, you're audible.
Yeah, thank you.
Yeah, it's my pleasure to also welcome the resource persons. Some of them are from England, some are from Sweden and other countries. And I hope that they will enrich our knowledge, the knowledge of the participants and people concerned with this theme of the webinar in the next 3 days. I welcome all the resource persons. I also take this opportunity to welcome Reverend Fathers, Rector Dr. Bernard Prakash Barnes, Reverend Father Maria Xavier, the Vice Rector, Reverend Father John Paul, who is the campus administrator, and who always have stood by all these activities they have supported, and I welcome all of them. Welcome you, Fathers. I take this opportunity to welcome Professor Othbert Pinto, the Director of PG Block, for this webinar. Welcome you, sir. I also take this opportunity to welcome the Vice Principal, Mr. Sanjay Nair, Mr. Prakash Kutina, the IQAC Coordinator, and all the other dignitaries for this program. And I take this opportunity to welcome all the faculty members of chemistry and allied sciences for this international webinar. Welcome you, sirs and madams, for this program. And I'll be failing in my duty if I don't do this. I welcome all the participants, teachers, researchers, students from St. Philomena's College as well as from other institutions to take part in this webinar. And I sincerely hope and pray that this webinar will enrich the knowledge of all the participants, all the faculty members about the importance of pharmaceutical chemistry and also the need of protecting intellectual property rights and the mode of doing so. So once again, welcome all the people involved in this and hope for a very productive 3-day sessions. Thank you.
Thank you, Madam, for welcoming all of us. And on behalf of all of us here, I welcome you to this webinar. We have an eminent person amongst us, Reverend Monsignor Leslie Morris, Episcopal Vicar, Educational and Medical Ministries, Diocese of Mysore. I request Reverend Monsignor to give the inaugural address.
Reverend Father Dr. Bernard Barnes, the rector of the college, Dr. Ruth Shantakumari, the principal, Dr. Alphonsus, the head of the Department of Chemistry, which is organizing this webinar, all of the dignitaries who are gathered here, all the speakers, the keynote address person, Professor Mahadev Swami, Mahadevan, all the other speakers. I'll be giving probably just a few introductory remarks about this whole webinar. The topic is something which is very large, pharmaceutical chemistry on the one hand, which is an ocean by itself, and the whole aspect about intellectual property rights, which covers not only pharmaceutical company, but every other field of human interactions and commerce. First, to begin with, pharmaceutical chemistry is something very recent, nascent one. Probably after the 17th, 18th century only we have it. But pharmacy is something age-old. Way back in the Middle Ages or even earlier probably, depending on the folk knowledge that was there, man had always ventured hit or miss kind of a system where medication was doled out. Various maladies of man. Most of the time they didn't even know what the cause was, but this was going on for a very long time. It's only after the 16th, 17th centuries that chemistry started entering into the field, and especially when people started finding alkaloids could have a special role to play in curing a lot of sicknesses and bringing back the balance in the human body. Human body by itself is structured in such a way to take care of itself, its own metabolism. But maybe as the time changes, as we become more and more advanced in our civilization with a lot of pollution, man's body needs external support for that balance. And it is this which has brought the pharmaceutical industry, pharmaceutical chemistry into play. For the first time, I think it is in 1727, man ventured into trying to use chemistry in pharmaceutical industry and tried to make a lot of things. Now, a lot of companies which are hitting the headlines today, including Pfizer, was started in the United States. The initial people who got into it are the Germans, who tried to use the chemical knowledge, the knowledge of chemistry in pharmaceuticals, and later in Switzerland, in UK, and other places. Pfizer, which is hitting the headlines today, especially with the COVID-19 vaccine, was started in America by two Germans. And in the beginning, pharmaceutical industry was anyone's field. Anyone could start something, anyone could try something. But then a revolution came when aspirin came into the picture, where people began to find the need for controlling something. Initially, anything could be bought across the counter, including citric acid for soft drinks and even heroin and other medicines. But the invention of penicillin brought about a revolution as well, because now people began to look at pharmaceutical industry not only to cure diseases of individual people, but large masses of people where life was at stake. Today we are experiencing that in the COVID-19 pandemic. If that is the history of pharmaceutical industry, pharmaceutical chemistry, on the other hand, intellectual property rights brings about very important aspect to control this. Because what happened in the beginning was the pharmaceutical industry didn't bother so much about the ethical aspects of it. But gradually man began to realize that someone need to take own responsibility for whatever is doled out as a medicine. And that brings a bioethical aspects and controlling the whole thing. And it is this which brings intellectual property rights into place. Because whatever is given cannot be given without any owning responsibility. Somewhere, someone has to try to bridge the gap between the two. And ultimately, there's much more important aspect of it regarding the whole thing. The whole pharmaceutical chemistry or pharmaceutical industry is for the wellness of human beings. Can wellness of human being be made a commerce, a commercial thing? But then it was, I think, a military man, Colonel Ellis in the United States, for the first time introduced R&D in pharmaceutical industry, research and development, because this was very essential, because everything could not be tried on human beings, but something had to be tried before it is used. And with that, the whole cost of pharmaceutical industry went up, and especially with the successive wars, the American war, then the First and Second World War, which needed a lot of antiseptics, painkillers, which made the pharmaceutical industry to become a commercial industry producing medicines in mass. Today, of course, we are caught up in a situation where the whole mankind is looking for solutions for the health of man, solutions for the survival of man, solutions for wellness, solutions for how best we can find solutions to make health a right for everyone. And I'm very happy the Department of Chemistry has taken this topic in collaboration with Visvesvaraya Technical Trade Promotion Center of the State Government of Karnataka to bring this topic for discussion, because this is apt today, and we need to have a lot of discussion. And I see a lot of speakers, people from the academicians in chemistry. There are CEOs of pharmaceutical companies. There are other people who are involved in developing, innovating the research and development, and also people from the law department, from the intellectual property rights, which probably will bring the ethical aspect to fore, make the pharmaceutical companies to take own responsibility. And I wish this webinar all the success. So the deliberations, I'm sure, will be very useful and helpful for all the participants, the teachers, the researchers, the students, and general public. I wish the webinar all the success. May this bring and throw greater light on this particular aspect and help us clarify a lot of situations, lot of aspects which are still in the state of doubt, in a very fluid state, especially in the context of the vaccine, which are the different vaccines in the United States, the Pfizer, whether it is Serum or whether it is Bharat Biochem or whatever Russia has done, where people will take responsibility not merely because the time is short within which a vaccine has to be found out, but that cannot give us a reason to do something where we can experiment with the lives of people. The biological aspect, the ethical aspect also need to be considered, and also the intellectual property rights. I wish the organizers of this webinar all success. Thank you.
Thank you, Reverend Monsignor, for your inspiring message. We have with us virtually our chief guest, Professor K.M. Mahadevan, former Registrar, Evaluation, University of Mysore, who will be delivering keynote address. I now request Dr. Ravi J.D. Saldana, Associate Professor, Department of Chemistry, St. Philomena's College, to introduce our keynote speaker.
Good afternoon, all. It's my great pleasure to introduce our speaker, Professor K.M. Mahadevan, the former Registrar Evaluation, University of Mysore, who is going to give the keynote address. A very special welcome to Professor Mahadevan for agreeing to deliver the keynote address and joining this online webinar today. I'm sure his address will make a lot of difference to the participants of this webinar. Our speaker today is a simple, young and dynamic person. He is an outstanding scholar, academician, and researcher. Professor Mahadevan has a brilliant academic record. He obtained his MSc degree from the University of Mysore in the year 1997 with a distinction, and in the same year, he cleared the national eligibility test conducted by UGC. After this, he joined as a research fellow at CFTRI Mysore in the year 1998. He was appointed as a lecturer at Kuvempu University in 1998, and later he obtained his PhD degree from the same university in the year 2003. He was then selected for the postdoctoral research program at South Korea under Brain Pool Fellowship Program in the year 2010. He was also a visiting scientist at University of Sunderland, London, UK. Starting his career as a lecturer or assistant professor, he has risen to the position of Registrar Evaluation, University of Mysore. In this journey, he has held many responsible positions, such as Head, Department of Chemistry, Kuvempu University, as chairperson to both BOS and BOE, coordinator for distance education, faculty advisor, and also as the director for PG Center at Kadur and Chikmagaluru of Kuvempu University. He was promoted as a professor in 2013. He later became the Registrar Evaluation of Mysore University in February 2019 and continued to be in office till recently. He has also been beyond the system, while there are places at the state level where he has served as scientific advisor since 2005 till date at Bioorganics and Applied Material Private Limited, Bangalore, Suman Laboratories, Shimoga, and Azyme Bioscience Private Limited, Bangalore. He has published more than 240 research papers and edited 12 books. He has guided 47 students successfully towards their PhD degree, and another 5 students are presently working under his supervision. In recognition of his research contribution in the field of chemistry, he has been bestowed with the Distinguished Industrial Research Scientific Award by Bioorganic and Applied Material Private Limited, Bangalore, and also by Mid-Career Award, prestigious award by the UGC in the year 2019. In addition to this, he has completed over 5 research projects funded by DBT, DST, CSIR, and UGC. And a few projects funded by DST and VGST are in progress. Some of his research investigations have become the part of the textbooks at the higher education level. Also published in foreign textbooks attribute high quality research work being done in his laboratories. As the Registrar Evaluation, Mysore University, since February 2019 till recently, he has proved his administrative acumen in effectively conducting the examination process and other relevant issues through innovative methods. He was a hard taskmaster. Currently, he is the Director of Distance Education, Director for Outline and Online Programs, Coordinator for Chief Minister's Kaushalya Karnataka Yojana Program. He continues to be an active person in contemporary research in chemistry. So, with this introduction, I am greatly honored to invite Professor Mahadevan for the keynote address. Over to you, sir.
So, thank you so much for your nice introduction. I'm really very much honored for being invited me to this webinar. So, very good afternoon to all present over here in this webinar. organized by St. Philomeno's College, Mysore, about pharmaceutical chemistry and intellectual property rights. And as we are facing since last 1 year, a pandemic situation all across the world, the only small virus is going to prevail, and it's going to trouble the entire world, all humans, and even including the animals. Recently, report says, and pandemic, this virus is going to attack everyone. I think it's entire living organisms in the beautiful planet, it is Earth. And see, as an organic chemist and as a medicinal chemist, and we have been working different diseases or targeting by different diseases since my insertion into the university as a faculty and as a researcher. And we engage with different funded project. And as a scientist and as a professors, what we noticed since last 20 years, and though we are working on medicines with a different uh, various diseases, and we couldn't able to get appropriate funding for continuing our research. So this was being happened for the last almost 20 years. And as a result, the situation all across the world, not only in India, the people, I mean, administrators or people who are in the power country, they are not in a position to fund for the researchers all across the world, particularly chemical biology. So chemical biology is a very important field. That's what we are facing now. And people are not immediately is going to give a drug, a small virus, for example. And though as a medicinal chemist, we have produced almost 50 therapeutic agent for viral infections, that is antiviral drugs, from 1949 to 2006, HIV. Like many viruses, about 50 antiviral drugs have been invented apart from vaccines, and they are working well. And these are the different two fields which we look into the antiviral molecules. One is a vaccine, another is a drug. As an organic chemist, we look into a drug. And when a molecule becomes a drug, it becomes a commercial product. Then all the sorts of legal obligations will come into picture. As a result, political willpower will decide the fate of many life-saving drugs. That's what now we are looking at intellectual property right. So with this situation, though we work very hard at the laboratory level to develop a new molecule for different life-saving drugs for different various diseases, and finally, so when it comes to a product, when we call it as a product, when it calls it a product, it will gain a commercial impact. As a result, the social impact and political impact will come into the picture. And though we work so many years to bring into the market a simple drug, a very beautiful drug, it could be life-saving, since what we have developed many drugs as a scientist. And finally, a political willpower is going to decide whether it can be used or cannot be used. That is a part of the intellectual property right. And this is a very beautiful and very interesting platform that we are going to provide to get aware of all the new researchers and upcoming researchers and young researchers, how these things is going to happen in the society with the people deal with intellectual property right, and a real scientist is working to tackle the scientific problems to keep everyone happy and do a comfortable life in terms of technology and in terms of keeping a good health of the whole everyone lives in this beautiful planet. And therefore, I congratulate the team effort by the St. Philomena College, which decides the title of the topic as pharmaceutical chemistry and an intellectual property right. I congratulate them for having a fruitful discussion with this webinar, and I wish them a very successful webinar. Thanks to Kunal.
Thank you, sir, for your inspiring message. We have with us virtually Professor Prabhavati Rao, Facilitator, IP Initiative and Geographical Indication Cells, Visvesvaraya Trade Promotion Center, VTPC, Government of Karnataka, Bangalore, to speak a few words.
Thank you, ma'am. On behalf of the Visvesvaraya Trade Promotion Center, Department of Industries and Commerce, Government of Karnataka, and St. Philomena's College, and all the wonderful stakeholders who have logged in virtually from different parts of the globe, a very warm welcome to what I would call a very timely topic, and I'm sure no one could agree more. Time is now, because we are, facing one perhaps one of the most challenging situations of our times, at least in our living memory, where COVID really struck us hard. But the good part is that all our brethren, all our stakeholders have come together on a common platform, ideating and trying to nurture the innovative streaks that we are all about as a wonderful nation. And today, as I represent the Visvesvaraya Trade Promotion Center, Department of Industries and Commerce, let me take a moment to introduce the organization I represent before I share a couple of thoughts and actually try to whittle it down in the short time that has been given to me. So, friends, not sure how many of you are aware that we are perhaps one of the very few exclusive nodal agencies in the country, in any state, that has an exclusive nodal export promotion center. You do have exports happening across the country, the length and breadth of the country, across the states, but then most of the places, one line in bureaucracy or one particular vertical is what exports is all about. But here we are, Karnataka, blessed with a nodal agency for the promotion of international trade. So our main charter is handholding budding exporters, promotion of initiatives and incentives in the international space through various schemes. As you all know that while exports is a central government subject, both the center and the state provide us with a lot of incentives and schemes to nurture this space. And that is perhaps, I would say, one of the reasons why I personally find a lot of youngsters, a lot of startups, and many others entering this field of international trade. Because in the lighter vein, I always say that typically long ago, the ancient kind of a thought that we had was that people who are entrenched in businesses, who are into, say, manufacturing or production, already have a business going about them, are the guys who would probably look at exports or as foraying into exports. But here is our generation who are trying to leverage the kind of ecosystem that has been provided by both the center and the states. And I find a lot of academicians, for one, a lot of startups, as I said, who have been coming forward to enter into this field. Now, coming to the intellectual property bit, I'm so proud to belong to a country and even to a state, actually, which has given several firsts in several spheres of the economy. And I'm sure all of you also would feel with a sense of pride that we are actually the innovation hub, one of the leading innovation hubs of Asia. And as per the latest Niti Aayog ranking on innovation, Karnataka stands first. Not just that, there are many, many more areas where Karnataka has given several firsts to this nation. We have been rated as the most dynamic city in the world. And when it comes to IPR, probably if I relate, we are the only city in the country which has its own logo, and that's BU. So that's IPR coming in play. I just heard one of our predecessors talk about, the traditional medicine, the ancient systems that we were all, so popular for. So, a few thoughts that I want to share. One of them is, my dear friends, let's be aware of the intellectual property rights that we have. Back in time, if India was not aware of its IP rights, we would have lost neem, turmeric, and all of those. I'm sure all of us know those stories, those very, very controversial stories where, and some of them were half baked. People were not aware of the second part of the story, meaning that, when countries tried to, when somebody XYZ tried patenting, let's say, neem or turmeric, which was India's rich repository of traditional knowledge. And that's an IP for you, IP tool. When XYZ wanted to do that, you know how India as a member of the WTO1 and as a member of the TRIPS, we tried to see how we could, invoke the provisions and ensure that neem and turmeric, rightfully, which belong to this country's repository of traditional knowledge, is, we get it back. And we fought, and those are the landmark cases. What else could you ask for? That is all because we were aware of our rights, my dear friends. So remember, one of the main thumb rules in the IP, ecosystem would be, please remember that we have a bundle of rights. It's not just about patents alone. It's patents, trademarks, copyrights, geographical indications, industrial designs, so many of these IP tools which we need to protect. I find a lot of youngsters coming forward with fantastic innovations. But the challenge what I find is that they probably are not aware of a very important element in the IP system and IP filing called as prior art. To just give you a quick example, here was this popular college in town which came up with a fantastic invention, and that was one of the inventions in the agrobiotech sector, which as a child I would always ask my mom, will somebody not come out with this? And here somebody did. Our college came out with that. But unfortunately, they lost the right to file a patent for that wonderful invention because they were not aware of what is called as prior art. Meaning, until you file your patent or any of the IP tools, please do not disclose it to the world. So here was this fraternity, which had put out in social media, which went viral. They put up on their website. They called the press and media to the campus, and they publicized about it before it got filed. So my dear friends, just remember this golden rule of the thumb. Don't make it what is called in technical parlance as prior art. So it becomes prior art. It's already established. It's already there. That is what the patent office will tell you. So first file, protect it, and then announce it to the world. That's one. Second important takeaway and thought that I want to always share is that let us respect each other's intellectual property. Let's not transgress. Today it could be me, it could be you tomorrow, your child, your offspring. It could be anyone whose intellectual property is being transgressed. I'm sure nobody appreciates that. Let us honor and respect others' creation and creativity. Let's honor them the rightful way. And there are many ways to do it. That's one. And importantly, remember, the timing of filing is very important. I also heard one of my predecessors, mention about how the Pfizer medicine, vaccine, and all of that, the thoughts were germinated way back with the Germans. This is precisely what has happened, my dear friends. Country ABC invents something, and country XYZ goes ahead, files the patent. That's all about this game. It all depends as to when you file. There's something called the right time for filing, and then, afterwards, when it gets published in the media, we tend to say, I've heard a lot of my friends say, hey, this was an invention which I was thinking about, I was about to file it. But then you have somebody else who has already gone ahead. So that is called, if at the sense of timing, if you really feel that something is very creative, you just go ahead, protect it. On that note, I would just like to remind you about examples of the very popular telephone, which we associate with Graham Bell, then Edison, the light bulb, the great giants, theory of relativity, Einstein, then, names like Tesla and Marconi associated with radio, and then Michael Jackson's famous moonwalk signature. All of these we associate with those respective icons and celebrities, right? But actually, if you go back in time to world history and the n number of examples, you will realize that before them, before these people went into the Hall of Fame and these particular inventions were associated with their respective names, there were a whole lot of people like you and me, the common man who probably thought that we could not be a next Einstein. They were the ones who had actually worked hard. But these are the set of people who became popular because they went ahead filing intellectual property right on time. And that's what matters, my dear friends. So go back to history, you'll find a number of such examples. So my appeal and request to each of you, dear friends, would be, we are abuzz with ideas. We are an epitome of that. Let's unleash our creativity. Let's do wonders for this wonderful nation called India and Karnataka. All of us, let's put our heads together, collaborate. This is the time. I think I'm short of time, otherwise I would like to go on and on. But on that note, let me wish the webinar all success, and with a wonderful lineup of speakers for the next 3 days, I'm sure each one of us would pick up useful gleanings on the subject and contribute our might to our motherland. Thank you so much.
Thank you, madam, for your enlightening talk. The essence of all beautiful art is gratitude. I now request Dr. Alphonsus de Souza, HOD, Department of Chemistry, to give the vote of thanks.
Good afternoon, everyone. No duty is more urgent than rendering thanks. Yes, every time we remember to say thank you, we experience nothing less than heaven on earth. It gives me great honor to thank Reverend Father Monsignor Leslie Morris, Episcopal Vicar, Educational and Medical Ministries, Diocese of Mysore, for accepting our invitation and inaugurating the 3-day international webinar and delivering inaugural address. On behalf of everyone present, extend a heartfelt thanks to you, Father. I am privileged to render special thanks to Professor K. Madhavan, former Registrar Evaluation, University of Mysore, and Director of PG Center, Coimbatore University, Kadur, for accepting our invitation and sharing your valuable thoughts. On behalf of Philo's family, thank you very much, sir. It's my honor to thank our guest of honor, Professor Prabhavati Rao, Facilitator, IP Initiative, Visvesvaraya Trade Promotion Center, Government of Karnataka. On behalf of Philo's family, I render sincere thanks to you, Madam. Great leaders don't tell us what to do, they show us how it is done. We have such a leader, Reverend Father Bernard Prakash, Rector Manager, St. PHILOMENA'S institutions. On behalf of everyone present in this webinar, extend a heartfelt thanks to you, dear Father. I render special thanks to Reverend Father Maria Xavier, Vice Rector, and Reverend Father John Paul, Campus Administrator, who are always spreading the fragrance of confidence and encouragement in the campus. Thank you, Fathers. A sincere thanks to our beloved Principal, Dr. Ruth Chand Kumari, who is an inspiring spirit behind all the activities of the college. Thank you, Madam. It gives me great honor to thank Professor Sanjay Nair, Vice Principal, Professor Othpot Pinto, Director of PG Center, Professor Agnes de Souza, Academic Coordinator, Professor Brito Dominic Ryan, Controller of Examinations, Mr. Prakash Kutinno, IQC Coordinator. Thank you very much to you all. I am privileged to render special thanks to all my colleagues in the department, both UG and PG, and all the members of Philo's family. Thank you very much, my dear colleagues. Special thanks to all the delegates, those who are present in this webinar. I wish you memorable time with us for 3 days. A word of appreciation to all the resource persons, those who have consented to share their intellectual thoughts with us. My sincere thanks to all technical staff for rendering technical support. Thank you, one and all. Have a pleasant time.
Participants for registering, and I request all of you to stay connected, making use of a stable internet connection throughout the webinar for all the valuable inputs given by our eminent speakers. Participants will be provided with an opportunity to interact with the speaker towards the end of the session. Questions along with the name of the participant and the institution can be posted in the chat box, which would be answered by the speaker at the end of each session. We shall now move on to our first session, Interesting Aspects of Fluorine Chemistry by Dr. Sridhara Bhatke, Technical Coordinator, ArcGen Pharma Private Limited, Bangalore. Thank you for accepting our invitation, and I'll welcome you, sir. I request Dr. Asha MS, Associate Professor, HOD, Department of Chemistry, PG Center, St. Philomena's College, to introduce our resource person.
Good morning, everyone. I feel elated and deep pride to introduce our speaker, Dr. Sridhara Bhatke, Technical Coordinator, Arcgen Pharma Private Limited, Bangalore, of this session to all who have participated in this three-day international webinar. Dr. Sridhara Bhatt holds master degree in organic chemistry and awarded PhD in heterocyclic chemistry from Mangalore University. He has got CSIR Junior and Senior Fellowships from Government of India. He has been renowned as guide for PhD from Mangalore University. He has got 20 years of industrial experience in the field of agricultural chemistry, pharmaceutical chemistry, polymer chemistry, and custom synthesis. Dr. Sridhara Bhatt has proficiency in research and development and manufacturing as per good manufacturing practice guidelines, active pharmaceutical ingredient regulatory requirements, intellectual property rights, process safety and hazard evaluations, technology development for API, agrochemicals, and intermediates. His achievements in various positions like as head API, R&D, Alchem Laboratories, Bangalore, which is the 7th largest Indian pharmaceutical company, having strong presence in India and abroad. Worked as Assistant Director in a Tata Group of Company, R&D, Advinus Therapeutics, Bangalore. Engaged various positions in R&D, Rallis Research Center, Bangalore, and represented the Tata Group company for R&D activities in India and abroad. Our speaker has published more than 22 research papers in national and international peer-reviewed journals and I'm glad to say that he is one of the international patents. I now hand over the session to our speaker, Dr. Sridhara Badke. Welcome you, sir. Now the session is yours. Sir, can you increase the volume, sir? Sir, can you unmute, sir? Sir, am I audible to you? Hello, sir?
Sir, we can't hear you. Sridhar Bhatt, sir, your voice is not audible. Can you please unmute?
Yeah, am I audible now?
Yes, sir. Now you are audible. Go
Okay.
ahead.
Sir, can you bring down
Yeah.
your camera, sir? Little.
Hello. Yeah.
Yes, sir. Okay, sir. Okay, sir. Done.
Okay. Fine. Good afternoon to everybody. Thank you, ma'am, for introducing me to the audience. At the outset, let me thank the organizers of this webinar, this St. Philomena's group, and this will be a wonderful opportunity to interact with right-minded people. At the end of the day, I think at least few good number of people will be benefited from this, and we can start interacting and we will get benefited. Now, let me move on to the subject. Today I'm going to touch upon the interesting aspects of fluorine chemistry. This is one of the emerging fields in pharmaceuticals. My talk is initially I will brush up on some of the important aspects of fluorine, then I will move on to pharmaceuticals and what is the impact of fluorine substitution on pharmaceuticals, what is the emerging field right now people are working on, and some of the applications of fluorine molecule. First, let me talk about fluorine. This is just to brush up on the youngsters, those are just into MSc. Fluorine is a compound which is represented by F and atomic number is 9. Its atomic mass is 19 and it is a lightest halogen. It is a diatomic represented as F2 and it is pale yellow gas and it is highly toxic. This one has to remember when we are working on fluorines. Now let us come into the reactivity aspects of fluorine. Fluorine is incredibly active. This reactivity is due to the electronegativity. We can see fluorine is sitting on the top of 17th column in the periodic table. The position is given to it because of its high electronegativity. We can see the atom of fluorine contains 9 electrons and one of the electron is unpaired and it tries to get paired and become neutral. We can see fluorine is represented as F2. Even though it is appearing as F2, it is normally not found because this F2 bond is not stable. Probably that may be because the atomic radius of fluorine is very small and there will be a repulsion between the nucleus. These neutron positive charges sitting in the nucleus. Therefore, this F2 atom is not stable. If we look at the old textbooks, we come across, F2 is not found in this form. It is always found in its third form, and normally it is calcium fluoride, which is abundant in nature and it is known as fluorspar. If you look at the abundancy of fluorine, it is around 13th most abundant element found in Earth's crust. Even though its abundance is so much, it is very rare to see it in the form of F2. Now, let us go back to the invention of fluorine. Before fluorine, HF, this hydrogen fluoride, was known. F2 came into existence. Basically, F2 is invented after inventing HF. Hydrogen fluoride is also a nasty chemical. People were trying to invent F2. While working on this, many people have lost their lives. This is because of the toxicity and the reactivity of fluorine. Finally, F2 was successfully made in 1986 by a scientist, German scientist, Henry Mosen, and he invented this by the electrolysis of HF, and he has used platinum apparatus for getting this F2. Prior to this, people are knowing the property of HF. They were using for etching the glass. So they knew HF cannot be handled in glass. That's why rest of the experiments were conducted in platinum or metal like platinum. As I informed, HF and F2 are toxic. For the discovery of F2, the scientist Henry Mosen was awarded Nobel Prize in 1906. But the pride of getting the Nobel Prize was not lasting for long. After receiving the prize, in 2 months, he lost his life. That is because of the toxicity of fluorine. This indicates how difficult it is to handle fluorine. Large number of people worked to invent fluorine as well as HF in 18th century, and we can get the information from the history that good number of people have lost their life. That's why the chemistry of fluorine is always challenging. Now, gradually let us move on to the pharmaceuticals. Normally we take drugs or pharmaceuticals to cure some diseases. Let us understand what are the drugs. Drugs are nothing but substances causing physiological effects when introduced into the body. These physiological impacts were done by the interaction of target proteins with the drugs. Let us look at let us look at some of the just wait for a minute. Let us look at some of the interactions and how the drug molecules are working. Anti-cancer drug which binds to rapidly dividing cells and make it to die. The drug activity is mainly targeting the rapidly dividing cells. Just look at the side effects of anti-cancer drug. That is, even the hair cells, they are also rapidly dividing. When you target rapidly dividing cells of tumor, they also target the rapidly dividing cells of hair growth, and it results in the loss of hair. This is the side effect of anti-cancer drug. So this tells us when two activities are having similar targets, if you target one, it is going to impact the other target also. So one has to be cautious while handling or while working on pharmaceuticals or inventing pharmaceuticals. Now we look at the natural abundance or naturally occurring some of the pharmaceuticals. In almost all the natural products or almost all the pharmaceuticals are having carbon-hydrogen bonds, but no natural product is having carbon-fluorine bond. Now, scientists are trying to introduce fluorine into pharmaceuticals, basically organic materials, so that we can get more efficacious pharmaceuticals. This introduction of fluorine to organic materials, organic molecules, is going to impact the properties of the molecules in an entirely different way. That is because carbon-fluorine bond is totally different than carbon-hydrogen bond. Now, before going to the next slide, I'll show you a small video which tells us how the drug is going to interact.
A drug is a chemical or biological substance that affects the body and its functions. Drug absorption is when a drug enters the bloodstream. The level of absorption can affect the speed and the quantity of the drug at the site of action. This is termed bioavailability. If a tablet releases the drug quickly, blood levels may become too high, whereas slow release may result in low levels of absorption. Additional factors affecting absorption and bioavailability include the properties of the drug and the physiology of the person, such as pH levels in the stomach and its speed of emptying. Therefore, specific formulations are used to release the drug at the desired speed. Common formulations include tablets, capsules, suppositories, transdermal patches, and solutions. Drug metabolism is the process by which the body breaks down drugs, which mainly occurs by means of enzymes located in the liver. Many factors, including intake of specific foods and other drugs, can increase or decrease the speed at which drugs are broken down, and thus determine their levels within the blood. A drug may be broken down to stop it from working. However, there are other drugs that need to be broken down or modified before they become active. These are known as prodrugs. Some drugs are directly eliminated from the body by the kidneys in urine. For this to happen, the drug must be water-soluble. Drugs that are not water-soluble may be metabolized by the liver or enzymes in the blood into breakdown products that can be eliminated in urine. Diseases that affect the liver or the kidneys can cause blood levels of a drug to increase.
This slide, this small video, has given us some inputs regarding how the drug molecules enter the body and what are the modifications it is going through and how at the end it is going to reach the cells. Now, let us understand what are the stages when we swallow in medicine, it undergoes and reaches into the cell or the active site. We eat food or we drink many things. Whatever gets into stomach, it is getting into blood also. That blood directly gets into liver. Blood from stomach and intestine, it passes through liver before entering into the rest of the body cells. That is because the food material or any other material what we eat, it cannot get into the body in the same form. We may eat many things as we like or as per our desire, but it may not be the desire of the body. Body has to be given the food the way what it wants and what it can digest. So this digestion is happening, and it is that particular food material or the particle which are going to get into bloodstream are degraded or metabolized, and finally liver is putting those metabolized thing into the bloodstream, and it further goes into different parts of the body, and those materials, broken down materials or the metaboloids, will be absorbed by the cells. And the same is the case when a medicine or pharmaceutical is taken into the body. It enters the liver and it is further broken down. Any drug which enters the liver is metabolized. This metabolism is nothing but breaking down of the material. What the material comes out of liver and into the bloodstream, it depends how active the material is, and the bioavailability of the drug depends on the metabolism of the drug material. When the drug is metabolized and further it gets into the bloodstream, and finally it reaches the cells or the target molecules where it has to reach. Now let us look at few things. That is the first fluorine molecule which was marketed and used in pharmaceutical industry. This is the steroidal molecule which is known as fludrocortisone. It is a steroidal drug, and you can see here the fluorine is introduced. Prior to it, this is the molecule which is known as hydrocortisone, which has no fluorine. The fluorine here is replaced, the hydrogen here is replaced by fluorine, and it totally alters the activity and bioavailability of the molecule. Let us look at the role of fluorine in pharmaceuticals. Any molecule to become effective or successful pharmaceutical, these are the requirements. The molecule has to be stable. It is not stable outside, it should be stable inside the body. It should be soluble so that it will be getting into blood and reaches the target molecules, and it should bind to the active sites. If these three properties are met, then the drug will be more successful. Now, fluorine is introduced to enhance the metabolic stability of the molecule. The metabolic stability is nothing but when the molecule is getting into the liver. If it is stable, liver cannot degrade it. It should be stable. If it is degraded, the molecule may or may not be active because its property is completely lost. Fluorine can modulate the physicochemical properties. Once we introduce fluorine into a molecule, it can completely change its physicochemical properties because the activity of fluorine, it can vary the basicity and it can vary the liophilicity. This basicity and liophilicity, it affects the bioavailability of the molecule. Fluorine can alter the conformation of the molecule, thereby binding affinity. Because any molecule to be bound to the target, it should have specific conformation. This conformation can be altered by substituting hydrogen by fluorine. Let us look at some examples. Ezetimibe, it is a cholesterol reducing or cholesterol lowering drug which is available in the market. Let us look at the pre-drug. It was the lead molecule here, what I am showing, SCH48461. You can see the fourth position here, there is no fluorine. it is attached to hydrogen. Normally, when a molecule, especially phenyl molecules, when they get into liver, the fourth position is oxidized. It is a labile site for oxidation. If it is oxidized, it may lose its property. That's why normally when this position is substituted by fluorine, the oxidation of this position in the liver will be stopped. Thereby, it enhances its metabolic stability. Look at this molecule and look the final molecule. Its metabolic stability has been increased by 50 folds. Here you can see the ED50 is 0.04 mg per kg, whereas when it is not substituted by fluorine, its ED50 is 2.2 milligrams per kg. Here we can see by the introduction of fluorine, the metabolic stability has been increased. Now look at the another molecule. This is silicoxib anti-inflammatory drug which is available in the market. Look at the pre-drug. This is the lead molecule where you can see the fluorine is at the fourth position and trifluoromethyl group is in the third position. The first molecule, that is the lead molecule, its T-half is 220 hours, whereas when the fluorine at the fourth position is removed, its T-half is 3.5 hours. In the first molecule, its metabolic stability is very, very high. That much retention time in the body is not required. If a molecule remains in the body for a very, very long time, it can complicate. So the requirement of the drug is when its job is done, it should get out of the body. Therefore, in some cases, it is required to regulate the stability, metabolic stability. That also can be done by eliminating fluorine. If the fluorine is there, it enhances the stability, whereas when the fluorine is removed, that also from the fourth position of benzene, it is susceptible for oxidation. The fluorine is substituted by methyl, thereby the metabolic stability has been reduced. Let us look at another molecule. This is 5-HT receptors, HT10 it should be. Basicity is definitely required for binding the site. You can see at the first molecule, the basicity is due to this piperidine ring. Its pKa value you can see here, it is 9.7. Due to its basicity, it is able to bind to the protein, target protein, but because of the high basicity, it fails in passing through cell membrane. It cannot penetrate the cell membrane. In order to increase its affinity as well as its bioavailability, people try to lower its pH. That is basically they try to lower its basicity by introducing fluorine. When the fluorine is introduced, the basicity of the molecule is reduced. It has come down from 9.7 to 8.7. When we checked the bioavailability, the second molecule, its bioavailability has gone up and it's around 14%. Initially, it was very, very low. Prompted by this, people further try to modify this molecule by increasing 2 fluorine atoms. You can see in the last structure, there are 2 fluorine atoms. Its basicity is reduced further from 8.7 to 6.7. What has happened here is its binding to the target is completely lost because of the because of lowering the pH. So enhancing the number of fluorine also is not very useful here. And when there was no fluorine, there also it was difficult. So optimally substituting and controlling number of fluorine atoms in the molecule, people could be able to get the right molecule. But still, this can be tinkered further so that its bioavailability can be increased. I told one more thing, that is conformation. Fluorine can alter the conformation of the molecule. I have taken this example, that is 4-bromo trifluoromethoxybenzene and 4-bromo trimethoxybenzene, where when trifluoromethoxy group is introduced, the conformation of the molecule is changed. It is not in the same plane. The trifluoromethyl group is slightly bulkier than trimethyl group, and when the trimethyl group is there, benzene and trimethyl group and oxygen, they are in the same plane. When trimethyl group, when methoxy group is substituted by trifluoromethoxy group, its conformation is altered. It is no more planar. That planarity of the molecule is lost because of the substitution of trifluoromethoxy group. So this indicates the presence of fluorine atom alters the conformation of the molecule. Once the conformation is altered, its efficacy or its binding property to the target molecule also is altered. So by varying the substitution, basically fluorine, we can alter the property of the molecule. Fluorine substitution also enhances the binding affinity. Here you can see this is the target molecule, which is in blue color, and the next one is a compound containing fluorine. You can see the molecule which contains fluorine at the 4th position of benzene, which is represented in green color, and it is interacting with the 2 carbonyl groups of the protein molecule. When there is a fluoromolecule, it enhances its affinity toward the carbonyl groups. If there is no fluoro, that affinity cannot be expected. So looking at all these things, we can say by substituting the molecule with fluorine, we can alter the properties of the molecule, and by modifying properly, it is possible to alter the pharmaceutical properties of the molecule. Now we will come into another aspect of fluorine that is very, very interesting and one of the emerging field, and lot many things are happening in this area. That is molecular imaging. Look at the molecule which is given here. Fluorine has one more radioisotope. Earlier we were discussing about fluorine having atomic mass of 19. Now there is one more isotope of fluorine which is having the atomic mass of 18. It is available in the nature, but its abundance is around 0.2%. The rest is 19 atomic mass only. And this molecule is not stable. It emits positrons and forms degraded product, and this emission of positron is made use of in identifying some of the happenings inside the body. Molecular imaging is one of the non-invasive techniques used to study how body functions in certain way. Without cutting open the body, we can see what is happening inside. For that, this molecular imaging is done. And the emission, positron emission, is used in scanning, and the emission is called as PET scanning. That is nothing but positron emission tomography. Look at the molecule given in the side, that is tagged with fluorine 18. That is the radioactive fluorine is tagged to the glucose moiety, and this moiety is called FDG, that is fluorodeoxyglucose. Let us see how this is used for detection of some of the diseases. Normally, PET scanning is done for detection of cancers. What is done is FDG, that is fluorodeoxyglucose, is injected into the body and the body is diagnosed. Cancer cells consume a lot of glucose because it rapidly divides and it grows. This rapid cell division and growth of tumor cells requires a lot of carbohydrate. It requires a lot of glucose. When we are injecting FDG, this glucose goes and accumulates around the cancer cells. Look at the structure here. The first chart, which was scanned on November 16th, and the second one was January 17th. The first chart, you can see there are few spots where FDG is concentrated. In the scan, we can see the FDG near the cancer cells. In the second scan, that is done after 2 months, we can see the distribution of FDG is more. In the first chart, there is no cancer cell in the liver, whereas in the second chart, we can see the cancer cells in the liver, in the heart area, in the throat area, abdominal area, and all. So this is possible by scanning using FDG. Now we can use the similar chart or scanning to check the efficacy of the treatment. Suppose if we interchange these two charts, the second chart if we consider before treatment and the first one is after treatment, you can see there is a reduction in the first chart and there is a number of cancer cells in the second chart. Over a period of time, if the cells are decreasing, we can say the treatment is effective. If the cells are increasing, the treatment is not effective, and doctors will have to think about some other way of treating this disease. So, by making use of PET scanning, that is also where F18 is used, we can come across how to diagnose cancer and how to look into the efficacy of the treatment. This is one of the applications. Let us look at another application, that is detection of Alzheimer's disease. Here also PET, that is PET scanning, is a powerful imaging technique that enables in vivo examination of the brain functions. And using this scanning, one can understand whether a patient is suffering from Alzheimer's disease or suffering from some other disease. You can see the three images. The first image is normal brain. You can see the accumulation of FDG almost all around the brain. In the middle one, mild cognitive impairment is seen. Here, the accumulation of FDG is little low. In the last one, the third image, we can see accumulation of FDG in the brain is partial. This is because some of the parts of the brain are shrinking, or some of the cells would have gone, cells would have died, and that part of the brain is not working. By using PET scanning, it is possible to understand what type of disease and where exactly the brain is impacted. So these are the few applications of fluorine molecules in today's medication. To sum up, what I can say is fluorine is highly reactive material. One has to handle with care. We have seen so many people have died in the initial stages. Having that information in hand, if you want to work on fluorine, we have to take enough care. Otherwise, it will be a problem for individuals, to the loved and near and dear ones, and for the society also. Fluorine substitution can alter the drug by altering its physiochemical properties. It is possible to alter the properties of the molecule by properly incorporating fluorine atoms. Fluorochemistry can be a tool to handle some of the challenges faced by drug discovery scientists. Thank you all. Back to the department.
Thank you, sir, for your enlightening talk. I now call upon Dr. Rasha to give an overview of the session.
Thank you, madam. Dr. Sridhara Bhatke, he spoke on the topic, interesting aspects of fluorine chemistry. Under this, sir has emphasized about the basic concepts of fluorine for the sake of degree and master degree students. He focused on drugs, pharmaceuticals. Under this, explained about anti-cancer drugs bind to rapidly dividing cells. Under this, he displayed very interesting video based on how drug is going to be interacted. He elaborated on fluorine in medicinal chemistry. Under this concept, discussed about role of fluorine in pharmaceutical with the illustration of few molecules. Very innovatively given the information about how fluorine can alter the conformation. Along with this, focused on molecular imaging, which is one of the non-invasive technique used to study how body functions. Sir has given very interesting applications like detection of cancer, Alzheimer's diseases, and finally concluded that fluorochemistry can be a tool to handle some of the challenges focused by drug discovery scientists. So, we are pleased to have your informative sessions today, and we need your kind support in our future programs also.
Yes, thank you.
Thank you, sir.
Thank you, madam, for the brief summary. Now moving on to the second session for the day, Relevance of IPR for Pharma Industry by Srimati Bindu Sharma, founder and CEO of Origin IP Solutions, Bangalore. Thank you for accepting our invitation, and I welcome you, madam. I now request Ms. Deepa, Department of Management, St. Philomena's College, to introduce our resource person.
Thank you, Bindu. Srimati Bindu Sharma, CEO of Origin IP Solutions. She is a postgraduate in microbiology, LLB from Bangalore University, and a registered patent agent of Indian Patent Office. She co-founded Origin in 2010, and with a team of experts from various technical domains, her company is assisting academic institutions, corporates with protection, monetization of innovation, and intellectual property. Her company, Origin, has been finalized in ET Now Awards of Leaders of Tomorrow 2013. Origin was also listed as Company of the Year 2014, Patents by Silicon India. Recently, she was listed as one of the top 100 most powerful women in law of VIPF. On Feb 1st, 2020, Origin IP Solutions LLP has been recognized by Niti Aayog Darpan, Social Talks, Government of India for excellent contribution in the field of patents, and Bindu Sharma has been recognized for excellent leadership in the field of patents. The certificate recognizes Origin for outstanding professional achievement, remarkable leadership, assertiveness, and integrity, all of which has made a good progress for India in the field of patents. She has been a speaker in more than 300 national and international conferences, academic institutions, and corporates. She has authored various books and articles, and 3 of her books have been kept in European Patent Office Library. I welcome you, ma'am.
Thank you, ma'am. Thank you so much for giving me opportunity to be here today. Before I start, just confirm if my voice is fine. Is it audible?
Yes,
Yeah.
ma'am, it is audible.
Okay, that's good. Okay, so thank you so much, and I will start. I'll just share my presentation. So please confirm if you are able to see my presentation.
It is visible.
Okay, fine. So, so I will be speaking on the topic, the role of patents and pharmaceuticals. So, pharmaceuticals have been since long time a very interesting industry when it comes to patents. There are lots of news about patented drugs, off-patented drugs, and compulsory license. And then pharmaceutical drugs also deal with human life. So there have been issues related to whether public health is important or patent rights are important. So overall, pharmaceutical has been a very interesting industry with respect to patents, and I'll take you through some of the important provisions that we have in Indian patent law with respect to pharma patents. So this is one of the statements by Albert Einstein, where he says that true sign of intelligence is not knowledge, but it is imagination. So what this means is that with knowledge, you can only reach from A to B, but with the imagination, you can reach anywhere. So imagination is very powerful, and imagination is something which is required to generate new ideas, and the new ideas are protected in the form of intellectual property rights. So there is a whole cycle of events, but end of the day, innovation is very important for every industry, every college, every institution, so that we generate new ideas, we try to solve the problems in new way, and this ultimately boosts economy and contributes to the progress of nation. Now, if I ask you a question that what are the most powerful inventions in the history, then some of the, and please feel free to type in the chat box whenever I ask you a question. So if you look at the most powerful inventions in the history, then wheel is something that always comes up as most powerful invention in the history, because wheel, ever since it was invented, the performance and functionality of wheel remains same, even though we have now more advanced composition and designs of wheel, but overall the functionality of this product remains same. Then we have nail as one of the most powerful inventions. Then we have compass, because before invention of compass, there was no way to find out which direction you are moving in. So this makes the product very useful today. And then comes the electricity, which again is one of the powerful inventions in the history. Internet, I do not need to explain you how powerful internet is. We all know that internet is a very powerful invention. And today, if you don't have a network connection for 10 minutes, you get restless. There is no network coverage, no internet. So internet has become such a vital part of life today that we can live without food for a day, but we cannot live without internet for half an hour. Wright brothers, I think they, as an inventor, they were the first one to imagine that human beings can fly. Before this, Wright brothers, nobody could even imagine that there is a possibility of human beings to fly. Steam engine again is one of the powerful inventions. Then comes this printing press. So printing press is powerful because before printing press was invented, there was no way to make copies of any work, and making copy was impossible. So it was only like, you could rewrite the document. And before printing technology was invented, even copyright law was not there, because there was no possibility of making copies of the work. And that's why we consider printing press as a powerful invention. Electric bulb is again, important and very powerful invention in the history. What this lady is wearing is a bindi, and it is called as life-saving dot. So what happens is that in Indian women, there is a deficiency of iodine. So what the bindi she is wearing has a layer of iodine in it. And once a woman wears this bindi for 24 hours, the requirements in the body of iodine are fulfilled. So this is a very beautiful, simple, and useful innovation. And this again is a very interesting, I must say, a very innovative idea. You would have seen some videos on Facebook about this particular product. It is called as water ball. Nowadays, you see that lots of pollution is happening because of water bottles. There was a research, and they found that Coke is the highest polluter of environment, and the most of the pollution from Coke comes from water bottle and Coke bottles. Okay, so this product is called as water ball. So the outer lining of this water ball is made of a protein, and one can eat the whole thing. So there is no need to have plastic bottles. So for environment, this invention is very good, and this is a very nice and simple invention. Then, in public places, the water gets wasted, and this is one invention called as water aerator. If you do not have this product in your college, you must have it. So water aerator, reduces the consumption of water by 40 to 60%. And this is like a must product for public places. Now, when we talk about intellectual property rights, so intellectual property is creation of mind. So when I say creation of mind, I can say anything as creation of mind, but this creation of mind should have value. So if creation of mind, your new idea, new design, new book, periodical, journal, your painting, your video recording, if it has value, then it is called as intellectual property. And once you register your intellectual property, it is called as intellectual property rights. When you buy a new house, you have to register it to claim ownership. When you buy a new plot or new land, you have to have registration of land done. Similarly, if you have new idea, you have to register it. And you have to register it depending upon the kind of idea you have. You have to register it in appropriate office, and we have various legal regimes where you can register your innovation. So we have patents, one of the most popular form of IP. So patents are granted for the new inventions, and one patent is valid only for 20 years. And after 20 years, it is like it comes in public domain. Your articles, books, periodicals, journals, they are protected as copyright. Compared to patents, the term of copyright is quite long. It's 50 to 60 years from the date of death of the last author. So copyright is quite long as far as the term of copyright is concerned. Then comes trademark, where the trademark means name of the product, like, Bingo Mad Angles, then you have Lay's chips, Coke, your iPhone, brand of your watch, footwear. So brand name is called as trademark, and the trademark is very, very important business asset. And the companies like Coke, Wipro, Infosys, the value of their trademark is phenomenal. So it is a valuable form of intellectual property. If any product design is good, so forget about the functionality, but if you have a toothbrush where you like design of your toothbrush, or you have flower vase, bed sheets, a television, your iPad, iPhone, where only even lampshade, watch. So design gives protection only to the apparent look of the product, or we can say aesthetic look of the product. Okay, and then in India we have protection for semiconductor chipset design also. Further, geographical indication is one of the most important and most interesting forms of IP. Geographical indication is the specialty of a place. So if you talk about Karnataka, then Karnataka has registered many geographical indications, and in fact, highest number of geographical indications. I'm sure Prabha would have spoke about geographical indication in the morning session. So I would like some of you to type geographical indications of Mysore here. I'll give you some example, but you type some of them. Then Mysore silk is registered. This is specialty of Mysore. Chanpatna toys registered. Bidri work registered. Then these rose onions, they are also registered. Then we have Mysore sandal soap, Dharwad peda. These are geographical indications from Karnataka. Then this kind of kasuti, Mysore Park is registered. Then this particular variety of betel leaves, elaichi, jasmine, sandalwood oil. These all are geographical indications of Karnataka. Let me check if any one of you have typed something. Can you type some of geographical indications of Karnataka? Or if you're from any other state, you can type GI of your state as well. If I talk about geographical indications from other states, then you have Banganpalli mango from Andhra, Nagpur oranges from Nagpur, you have apple from Jammu and Kashmir, Himachal Pradesh. Kerala has registered a wide variety of rice. Handicraft is also registered. So geographical indication, that's in that way gives recognition to the states for their specialities. Trade secret is a form of intellectual property which is granted to the secrets of business. Like Coca-Cola composition is a secret. So this secret is a very valuable secret. So any secret is secret only till it is not disclosed. So if it is disclosed, then it is no more a secret. So some more examples of trade secret are McDonald's, Aloo Tikki, then MTR masalas, then various ketchups. So food industry specifically is full of trade secrets. Then in India, we have protection on traditional knowledge also. When we say traditional knowledge, it is the way a prayer is made. Then it could be a folk dance, folk song of a state, method of making carpet, method of making ardas in the Gurudwara, then acupressure, acupuncture, Kerala massages, folk dances of various states. Then you have puppet dance, puppet show, sorry, and Sufi music. So all of them, they are traditional knowledges of a country, and traditional knowledge is never registered, but traditional knowledge is protected only by documenting it and publishing it. Now, coming to patent, what exactly a patent is? So, in very simple words, patent is either a product or a process which has inventive step and which has industrial application. So, patent is a statutory right over the invention for a particular period of time, and patent is valid for 20 years, and after 20 years the patent comes in public domain, and then the patent owner cannot claim any kind of rights over the patent after 20 years time has expired. Claims of a patent define scope and kind of rights it has. So patent rights are territorial. That means that Indian patent is valid only in India, and if one needs a US patent, then one should file a US patent separately, but the patents are granted from country to country. The first patent in the history was granted to a German engineer in Venice, 1323, for a model grain mill which could cater storage needs of entire Venice. 1787 was the time when in the United States first patent was granted specifically for grain elevator. Compared to other countries, in India, we enacted patent statute quite late in 1857, but after independence in the year 1970, we had a proper enforceable patent act in place, and we are following the same patent act even today, even though with lots of amendments. This was the first patent granted by Indian Patent Office, Pankha pulling machine. So in the old movies, you would have seen this kind of piece of cloth in the middle, and on either side you have a pulley, and there is a person to pull this pulley, and when the pulley is pulled from both the sides, then this piece of cloth moves and it serves purpose of a fan. So that's why it's called as pankha pulling machine. So this kind of drawings are submitted when a patent application or patent specifications they are submitted. And in India, you can file your patent application either in Hindi or English. Both languages are acceptable. There are various reasons that people file for patents. For universities and educational institutions also, there may be various reasons to file for a patent. So the first reason to file for a patent is branding. It helps you brand your institution. Now, if you look at MIT University, then MIT University they have been filing close to 1,000 patents per annum, and their name is published in annual report of patent office. So academic institution can really use patents for branding themselves, and if the quality of patents is good, it can turn out to be a source of revenue for the institution. For the students, if they have patent in their name, it could make their resume more impressive, and there may be more interest from the industry also for placement of students. After filing a patent application, you can talk about it. So disclosure becomes very easy. So if there are companies like startups looking for some funding, so after patent application is filed, then talking about or disclosing details of the invention becomes easy, and one can better interact with the investors. As soon as a new idea is conceived, it's important that initial assessment of idea is done. So when I say assessment of idea, it means that you should check whether you are the first one to do it or somebody has already done it. So there are various ways you can check this particular thing. And if you do assessment of idea right in the beginning, then you can avoid reinvention of the wheel, and you can really save money, time and energy in the process, and you will have a better output, because patent filing till grant is a long process. So if you have not done your initial due diligence properly, then later on it will be more of a problem for you. So while performing a search to assess novelty of your idea, you must go through the existing patents, existing paper publications, and existing products in the market, and then analyze whether your idea or your invention is really novel or not. If somebody has already done it, then there is no point filing for patent again for the same thing. So in the invention, novel elements can be identified. You can always number them. And every invention revolves around some technical difficulty. So find out what technical difficulty is there, and the kind of your solution to solve that problem. And also, I'll take up this later. Inventions not patentable. I'll cover it later. So So you must search for novel elements of your invention and find out if the invention is novel or not. These four are the free platforms where one can perform a patent search and find out if the idea is novel or not. eSpaceNet is eSpaceNet is one. Then Google Patents is the second platform, which is free. Then USPTO maintains a database which is free to be used. Then WIPO has a database called as Patent Scope, and this is also like free. So these four databases are more than enough for you to do initial due diligence and find out if the idea is novel or not. There are three things that are required to be there in a invention to get a patent. So invention has to be novel, it has to be non-obvious, and it should have industrial use. So I will explain you very briefly one by one. So when we say novelty in the invention, so in order to get a patent, invention has to be novel. That means there should not be any paper publication, there should not be any patent, or there should not be any usage. So if there is no publication about the invention, then you can assume this invention to be novel. What this doggy is wearing in this picture is called as doggles. So because these so they have taken the word from goggle, so it is for dog, for it is called as doggle. And this company Ray-Bans is making this for the doggies, which protects their eyes from dust and UV rays. And for academic institutions like yours, it's very important to note that don't publish before you file for a patent, because publications they are very important part of career in academic institutions. And there are so if you are planning to have a paper publication, and if invention has good potential, so I will highly recommend you to file your patent first, and then you can make paper publication. This is a case for Mento versus Mentmore, in which the inventor they released the product in the market and then they filed for a patent. But then on the date of filing, their invention was not novel because the product was already in the market. So US Patent Office refused them a patent, saying that on the date of filing, your invention was not novel. So it is important that if you are planning to go for a patent, then don't make any kind of paper publication till the time you have your patent filed. And after filing your patent application, you can go for paper publication anytime. So if your invention is novel, then the second criteria to be fulfilled is that inventive step has to be there. So you cannot have a novel invention just combining two things. And I cannot combine my laptop and pen and say that I have a new combination and I have new product for which I should get a patent, because pen is a known product and a laptop is also a known product. So when you are filing for a patent, your invention should take technology one step ahead, and just mere combination of two existing products cannot be called as a new invention. So the image that you see here is torch, which that is integrated on the helmet, and this could be used by people working in the mines. So torch is known, helmet is known. So it is just combination of two existing products. So it may not be patentable. On the other hand, if you see that some people may find it good invention to be patented, the reason is that in order to integrate torch on the helmet, it is not just the simple integration of torch, but it has various angles which help the light focus on a particular point, which is important for people working in the mines. So opinion on obviousness or non-obviousness may vary from person to person. So one may find it to be obvious and another person may not find it as obvious invention. Third condition to get a patent is that it should have industrial application. That means the invention should have capacity of being used or made in the industry. So most of the inventions, they have industrial application. So there are very few patent applications that have been rejected on the basis that they lacked industrial application. So there was one case of John Lahiri Khan. In this case, invention was rejected a patent because the claims of the patent talked about novel concept of making friend. So the examiner said that novel concept of making friends is not invention because it cannot be made or used in the industry. So these three conditions of novelty, industrial use, these three conditions are required to get a patent. So once you have these three conditions in place, most likely you will get a patent. And there is one more important parameter to it, that even if these three things are there, but there is a class of inventions for which patents are not granted. I'll take you through them very quickly. Okay, so but otherwise, these three conditions are required to get a patent. So just to give you simple examples, so carrying water is difficult in developing countries. So this image that you see here is a water tire, wherein carrying water is easier. I will not say that it is a long-term solution for water shortage or carrying water, but yeah, for time being, I mean, it serves a purpose, and it's definitely better than carrying water or carrying pitcher on the head. So this water tire is a useful invention that way. What this lady is doing is she is drinking water from a river. In developing countries where potable water is an issue, this is called as water filter, wherein there are layers of filter, and any water you drink through this particular pipe becomes a potable water. And this again is a beautiful invention, simple and easy to carry, and it's a nice, useful invention. There is shortage of space in the cities these days. So this is a vertical garden. So the best part of this vertical garden is that excreta of fish, because down there is an aquarium and then you have a vertical garden. So excreta of fish is taken by the plants as nutrients, and whatever is rejected by the plants is taken by fish as a food. So this is a complete ecosystem in itself, and I feel it's a useful invention. Now, just because only three things are required to get a patent, so sometimes very funny patents get granted. So you can always check on web weird patents or funny patents, and you will come across many unusual or very strange patents that have been granted, and you always wonder how examiner could grant such patent. But the thing is, once these three conditions are fulfilled, then examiner has no reason to reject a patent. Now this picture is umbrella for the beer bottle. So it is a very interesting invention. Now what this person is wearing is not a mask. It is a device to control your weight. So if you are feeling hungry, you can put this kind of shield, face shield on your mouth, and you can see lock also here. So you can lock your mouth, and whenever your desire to eat food has gone, you can remove it. So the title of this invention is device to reduce your weight. This product is a diaper for the birds. So if you have diaper at home, then you can always buy if you have birds at home, and you can always buy diaper for your birds, so that they don't spoil the place, and what this boy is holding in his hand is a laser. So he can train his cat to follow the laser. So the title of this invention is device to make your cat exercise, because wherever you take this laser, cat will run, and this will be helpful to make your cat exercise. For the ones who have pets at home and pets at home with long ears, then this is a useful invention, because this is good for managing ears of your pet. You do good work in life, but people don't appreciate. So if this is the case with you, then you can you can have something like this. You can put this particular product on your shoulder and say well done. You can see that there is this wooden part that can pat your back and say well done, and you feel really good about it. And this is umbrella cum raincoat. This is most interesting invention. The title of the invention is collar for the snake. So if you have snake as a pet at home and you want to take your snake for morning walk, then probably you need this collar for snake. So this collar for snake is used to make your snake more comfortable for morning walk. So this again is umbrella for your pet. If it is raining, like these days Bangalore is raining almost every day, so if you want to take your doggy outside for a walk, then this is the product you need. So the whole idea of showing you all these examples was that once these three conditions of getting a patent are fulfilled, then there is no reason for the examiner to refuse patent, and patents are granted, and sometimes very funny inventions get granted. But if you have good ideas, don't ever think that it is too simple, because simple inventions also get patents. So you should not be simplicity of the invention is not a bar. Before filing for a patent, analysis of idea or due diligence of idea has to be done, followed by documentation of the invention. Documentation is very important because you have to submit very detailed document to patent office. So if documentation is not proper, you may have lots of objections later on saying that data is not complete. So it's good to give good documentation right in the beginning. So after documentation, you can file your patent application in India. Indian filing is almost mandatory. You must first file in India. And after Indian filing, you can go for foreign filing anytime if required within 12 months of time. Filing for a patent, first of all, documentation, and there is no way out, but you have to document it. After documentation, patent search should be done, and after patent search, the process of drafting and filing application has to start. Indian filing has to be done, followed by foreign filing. And after filing is done, it is important that since you have invested money in patent filing, so valuation of technology should be done, and then you must start working on commercial exploitation of the invention. Then infringement risk analysis becomes very important before product is launched in the market. There are some inventions that are not patentable in India. So funny inventions which claim anything contrary to well-established natural laws is not patentable. Any invention which is harmful for animal, plant life, or plant health is not patentable. You cannot patent the better method of polluting lakes, method of polluting air. So just discovery of scientific principles is not patentable. Then new property of a known substance. If substance is already known, you have just identified a new property, you cannot patent it. And then admixtures are not really patentable. Now, if I combine two existing products, like if I combine chair with umbrella, so it is just arrangement and rearrangement of two existing products, which is not patentable. Lots of methods of agriculture and horticulture, they are not patentable. Computer programs in India, they have been a very big question mark whether we should grant patents on them or not. Then mathematical method, business method, algorithms, they are not patentable. Plants and animals in the whole, they cannot be patentable. Literary, dramatic, musical, and artistic work is protected as copyright. Then scheme or rule, method of performing a mental act, method of playing game, presentation of information, topography of integrated circuit, then traditional knowledge, anything related to atomic energy, all these things are not really patentable in India. If you file patent on any of such things, then there is a possibility that you will not be granted a patent in India. When it comes to pharmaceuticals, Section 3D is a very talked about section. So with respect to pharmaceuticals, there have been lots of discussion on why India has Section 3D. So in very simple terms, I will explain you what this section says. It says that if alpha form of a compound is known, then beta cannot be patented. That means if a main molecule is known, then any derivative of main molecule cannot be patented. So there have been lots of objections with respect to Section 3D and in international forums because India has been pharmacy of the world, but still Section 3D prevents many patents because Section 3 does not permit patents on derivatives of the main molecule. So if one molecule is known, then esters, alcohol form, isomers, then combinations of such main molecule is not patentable in India. There has been a very famous case of Novartis. The drug was Glivec. So Novartis was selling this drug Glivec at a very higher price. It was 1,20,000 rupees per patient, wherein a generic version was priced at about 8,000 rupees in India. And India refused patent on this. The reason was that one alpha form was known, and then beta, Novartis filed patent for beta form. And then Indian Patent Office said that according to Section 3D of Indian Patent Act, they refused patent on this because it was new form of the same substance. So there has been lots of discussion about this Novartis case on international forums. So there were Cancer Patient Aid Association also, they filed opposition to grant of this patent. So in 2006, Chennai Patent Office rejected this patent for the first time, and it was a very big attack on ego of such a big pharmaceutical company. So Novartis then went to Chennai High Court, and further they went to Supreme Court also. But finally, Supreme Court also rejected their case. And when this judgment came, the world was watching this judgment of Novartis case, because this was a very crucial case in the history of Indian Patent Act. And India did not compromise with its policies. And India said that public health is more important for us compared to the patent rights. And this patent was finally refused, and it was granted all across the globe, but they could not get patent on this drug Glivec in India. Now, if I take you through the process of patent filing till grant, so patents are first filed in India. Then if foreign filing is required, then it should be done within 12 months. And after filing, the application is published. And once the application is published, then it is examined by the examiner in patent office. So there are qualified examiners in a patent office who, examine thoroughly the subject matter, scientific domain of the invention, and then the patents are granted. And in order to keep patents in force, renewal fee has to be paid from time to time. If renewal fee is not paid in time, then the patent gets lapsed, and the patent holder may also lose rights over the patent. So from filing till grant, and then renewal has to be paid in time. If I take you through some of the news, you see lots of news on pharmaceuticals, and pharmaceutical is one of the most happening industry with respect to patents. So these kind of news you see every now and then. Delhi High Court barred Glenmark Pharmaceuticals from selling the drug Zeta on the ground that they infringed the patents of Merck. Then Novartis filed lawsuit against Cipla from making a generic version of the drug of Novartis. Then Glenmark Pharmaceuticals was infringing 2 patents of Cymid. Then Novartis was sued by Intas Pharmaceutical for patent infringement for particular drug. And pharmaceutical is one industry where patents actually live life of 20 years, unlike other industries where the patents become almost redundant after 2 to 3 years and new technologies come. But in pharmaceuticals, patent live a good life of 20 years, and that's why lots of litigations come from pharmaceutical industries. So in order to avoid such infringements, it's important that, apart from protecting own rights, companies should take precautions not to infringe upon third parties' IP rights. The patent gives a set of rights to patent holder that, if a company has patent, then the company can prevent all others from making, using, selling, offering for sale, and importing such invention in the territory of India. So if there is an Indian patent with the company, so that company can prevent everyone else from making or replicating the invention in the territory of India. So since patent rights are territorial, so if there is an Indian patent, it is valid only in India. If it is only a US patent, then it is valid only in US, and people in rest of the parts of the world can use it without permission of the patent holder. There is something called as clearance search that is required to be performed before a new product is launched in the market to avoid the risk of patent infringement. This was one case of SmithKline Beecham and Fujimoto. This was a case that was decided in Tokyo District Court 1998. SmithKline Beecham filed a process patent in Japan, and this patent was granted after 8 years, and it was valid till 1993. But during the time when patent was in force, Fujimoto imported the infringed product and sold 68,000 tablets in Japan. So this was infringing the patents of SmithKline Beecham, and Fujimoto also manufactured. They imported and they also manufactured generic version of the drug. So SmithKline they filed a lawsuit against Fujimoto for patent infringement. But Fujimoto in the court, they argued that the process was not covered by SmithKline Beecham. But again, in the judgment, huge amount was granted as damages to SmithKline Beecham, and it was one of the largest awards in Japanese patent infringement cases. So it is very important that the due diligence to avoid infringement of any third party's IP rights is performed well in time, so that companies, they do not get into this kind of dispute. Then I will take you through this. Was a case in Africa. So in Africa, there was a time when lots of patients were dying of HIV/AIDS, and the drugs for the treatment were very expensive. So many multinationals were selling this drug in Africa, and the price of drug used to be $10,000. So $10,000 in Africa is a very high price. So there were patents also for the drug. So in this process, our Indian company Cipla, without bothering for the patents, they manufactured the drug and started supplying to Africa without caring about the existing patents, and they were selling it at a very cheap price, just 3% of the price. So African government also, they liked the idea, and they started procuring drugs from Cipla. And once the drugs were supplied, then there was lots of a discussion about it in public forums, and there was a meeting in Doha. India for supplying drugs to Africa when there were patents with multinationals. And then in Doha Declaration, it was concluded that if there is a difference, if there is a balance to be maintained between public health and patent rights, the priority has to be given to public health. You cannot let patients die because they cannot afford the drug and just enjoy your patent rights. So if there is a need, like this was the time of COVID, so if there were any important patents that were coming in way to manufacturing the drug, then government would have taken the charge and permitted some generic companies to manufacture the drug at. So there is a very interesting concept called as compulsory license in India. Most of the countries they have today. So compulsory license in very simple terms means that if there is a patented drug which is not available for people to use, and if there is any pandemic or epidemic kind of situation, then without bothering about the patent rights, the government will take charge and permit the company company who is interested to make the drug to make it affordable and reasonably priced for the consumer. So this concept is called as compulsory license. This compulsory license is granted by controller of patents. In India, the first compulsory license was granted to NATCO for Bayer's cancer drug. So patent holder Bayer had not met reasonable requirements of public. So if this happens, then government has to take charge and grant compulsory license so that the product is available to public at reasonable price and reasonable requirements of public are met. Now, patent filing till grant is a very expensive and time-consuming, tedious process. So it's important that once a patent is filed, then it is also commercialized in the market so that so that the money is generated. Thomas Alva Edison said that anything that would not sell in the market, I do not want to invent, because sale is proof of utility and utility is success. So whenever you have new idea, you must try hard to sell it in the market. There will be companies who may be interested in taking your idea and manufacturing the product. So there are lots of steps before the value of patent is performed. Now, in Mysore, you have a plot, then you have a plot in MG Road, Bangalore, you have a plot in Chikbalapur. So the price of same sized plot will be different from region to region because there are various factors that determine, price of the land. So similarly, price of the patent also depend upon lots of factors, and there are techniques to determine what is right value of the patent. So after value of the patent is determined, then it is important to connect with the potential buyers, and then the deal is signed. There is a amount that both parties agree to. Then agreement is executed, and that's how the product is brought in the market. So I'll just give you some of the examples. We have around 10 minutes now left for me to close session. Maybe we'll have last 5 minutes for discussion. So NDRI, National Dairy Research Institute in Karnal, they had this technology of low cholesterol ghee. So they signed agreement. They licensed this technology to a dairy in Patna. And this dairy, Patna Dairy Project, they bought the technology and they implemented the technology, and they made this low cholesterol ghee. And in the process, NDRI probably made good money. Some of the technologies, they are very good, but they don't sell in the market. But some of the products, they are very simple in nature, but they do very well in the market. Example, one of the most beautiful examples is Post-it. So Post-it was a good invention. It actually helped company in multiple ways, and they really had good sales right from the beginning. And the unique part of Post-it is that the composition of glue is such that you stick Post-it to any surface, and when you remove the Post-it, surface is not damaged. So this is beauty of Post-it. And then another successful product in the market was Snuggie. It was a fleece blanket, and this blanket had sleeves. And this product did extremely well in the market, and this guy made lots of money because of licensing this particular product. Then Koosh ball, very simple product. Most of us, we have played with this Koosh ball. So inventor tied few rubber bands together, and it finally was given a shape of a rubber core. But he never knew that this product will sell extremely well in the market. So this was also a very successful product, success story. So now you see that when you have new idea, due diligence is important. After due diligence is performed, then it should be filed as a patent. So as soon as your patent application is filed, you should start working on the commercial aspects of it. And in the due course of time, you see that once you sell your technologies or patents to potential buyers, you will be able to make money for your institution and for you in the process. Now, the question arises that, I've told you so many things and so many things to do, how to start it? So the first step that the institutions can take is training of faculty and students. So since you have taken this step already, do not stop here, but make this process continuous, have more and more of training in-house, as well as you can make outside speakers like me and Prabha. You can call us anytime. Within the institute, you can have IP audit kind of thing to see what are the innovations already available. You can document all the innovations and select the good ones. So good ones can be filed as patent, copyright, and design. When you have IP and you start the process of IP protection, you need to have IP policy and IP processes in place. In fact, foundation day of your college should be made as Innovation Day. So that you can celebrate as Innovation Day. And on this day, you can invite applications from your students, let them present their idea, let them talk about their idea. And you will see that, with time, you will start generating repository of ideas, and anytime you can select good ideas and file IP for it. After you follow these steps, quality of in-house research will improve automatically, because once you file your first patent, you get to know what is required to get a patent, novelty, industrial use, non-obviousness. So your research and the steps of research, they fall in place automatically. So this helps institutions in big way. Branding of your institute becomes better. People start recognizing your institute, because when your application is published, you can always share it with everyone. You can put on your website your corporate presentations, and you will have students that are industry ready, because once you have students who hold patents along with the institute, then there is lots of interest from the industry also. Placements will be better, and on the whole, you will start saving money and time. So all these things will happen only after you have good policies in place, you have good processes in place, and most important is you have good quality of innovations. And quality of innovations, there are I have some tips to share with you how you can improve quality of innovations. So from time to time, we bring very good videos for students and industry. So I will keep sharing it with you now, and you can go through them. We have a very happening YouTube channel. We publish lots of articles on our blogs. So you are most welcome to access all of them. And we have a free course on IPR also. It's a small course, and if you want, I can share link with you. So those of you who are interested can take up the course. It is free of cost, but yeah, after doing this course, if you want advanced version of the course, then that is paid one. But yeah, first level course is free. So here I end. I think I finished well in time, 4:25. So 5 minutes I thought we'll have discussion. So if you have any questions, you can just type it, or you can also unmute and speak. So I'll stop presentation, and we can discuss if you have any doubts.
Thank you, ma'am. Let me sum up whatever ma'am has just spoke. She spoke about various inventions. Very interesting facts were revealed to us. Life-saving dot about the bindi, which gives the required iodine. the water balls, the water aerators. The whole session was very interesting, wherein she also spoke about the product that were registered in our own state, that is Mysore Pak, or let it be Mysore Silk, or the beetle leaves, or the Dharwad Peda. All these things are patented. So, the journey of the whole 1 hour, it was really very interesting. She also spoke about the brief history about the patent, why the patent should be filed, and what the patents can protect, and the conditions for patenting. And she also emphasized that before publishing any paper, it has to be filed for a patent. So the type of products that are patented, she came out with various types of products also which are patented. It may be a wooden, a back tap that we can do it for ourselves, or a diaper for the birds, or the umbrella for the pets. It was very interesting. She also said that the idea is more important, wherein the emphasis was given for the idea. The details about how to file a patent and what are the rights of a patent holder was also discussed. There were various cases that were discussed for the R. The Novartis Glivec case, the Smithline Beecham pharmaceutical company case, the African HIV/AIDS drug crisis case. So all these gave us a lot of inputs for the day, ma'am. Thank you very much, ma'am, for the session. It was very informative, and hope to connect you in future as well. Thank you
Thank
once
you
again.
very much. Thank you so much for it. Thank you. Good day.
Thank you.
Thank you.
Thank you, Madam Deepa, for the overview of the session. May our bonds be as strong as chemical bonds. With this quote, I would like to conclude our today's sessions. Hope to see you all back with the same enthusiasm for tomorrow's session, which will start by 2 p.m. Thank you. Have a great evening.